maximum 30 minutes to give you a quick overview over the infrastructure and its role in supporting big data research in marine biology. So this is also a little bit of an advertisement talk on the Swedish biodiversity data infrastructure, which was only recently launched, but it has a 10 years or even more background. But to go a little bit further back in time, I want to just to talk a little bit about the issue where this big data challenge is coming from. If we're looking at the historical view or the historical perspective on how data are collected, and there is, of course, has been a continuous increase in the technical capabilities in the last 100 years. People used to go out sampling by hand, and today even biological sampling takes place more and more in an automated fashion. And the same thing happens also with the access range, that the technology allows us today to access almost any corner of the oceans with all kind of devices that you can either mount on a ship or launch directly from land. 100 years ago, most of the observations, actually biological observations, were coming from places just around marine stations. So in those days, the marine stations were the actual hubs to collect data, but today there's many more of such hubs where you can collect data from the ocean. And and that is, on one side, the technological development that has been incredibly fast in the last 20 years. But on the other side, also the demand for biodiversity data is increasing a lot. You see that on this slide, that this is the biodiversity data that is provided by the Global Biodiversity Information Facility, that is continuously increasing in terms of the papers that are published and the records that are used for this. Just to give you a few numbers how Sweden contributes to that, that's about 10 billion Swedish data records are downloaded every month. That makes every biodiversity data record, which is usually a species observed in a place and in a time, is downloaded on average 3 times every day. So that potential to collect data, once you collect them and make them accessible, to the potential to reuse these data is really, really high. And these data are reused in all kinds of different contexts, for basic research, for applied research, for policy support. So that's where these data infrastructures come in. Now there's just one little problem, that most of the data that we have accessible in these large databases are terrestrial data. So the terrestrial data, of course, are much easier to collect, and that is the challenge that we have in the world of marine biodiversity research, that we want to keep up with that big data delivery flow into the databases on the marine side as well. And how are we going to do that? Well, now marine biodiversity research historically has been, just like any other research, relying on conventional data collection, and that is often you're observing a species or several species in a community or in a habitat, and you have the species, you have an abundance estimate, and then you have a space and a time associated with it, and then you analyze that. And that may not be ever enough to if we want to collect only these data, it may not be enough to really start understanding processes such as global change and human pressures on the marine ecosystems. So what we really need to do here is we need to upgrade our methodology as marine biologists, and we have to rely on these new data sources that are coming from machines. They're not generated by humans themselves anymore. So here you see some examples. There are many more, like satellite and remote sensing, genetic data, sensor data, and even citizen science data are the data that we need to focus on now and collect. But they are these new type of data, they're a little bit different, because the raw data themselves are not associated per se with any species name. So what we often interested in is a trait or a character or a name of a species. And these data usually don't deliver that. They deliver images or genetic sequences or these kind of raw data on which we have to do some analytical processing until we get to the species name or to an associated trait that we are interested in, a biological trait. And that is what makes these data more complicated. And I will give you some examples of this. So what is important here, that the data structures that we are providing and building up and are now making such data also accessible to researchers. So I guess the most of the marine biologists or actually have been using or want to use or currently use some of these data, but often this is very individual projects, and the data in a way that is often, when it comes to the publishing of the data, it's more research focused, and the raw data is not always completely accessible. So that's my point here, that in order to make these data really reusable and findable for other researchers in the same way as we've done this for the conventional biodiversity data, we have to upgrade also the infrastructures to be able to provide these services and these functions. So now we're coming to our infrastructure, the Swedish National Infrastructure for Biodiversity Data. It's called SBDI, the Swedish Biodiversity Data Infrastructure. If you go on that link that you see there, you can get to the website, and there is that is an infrastructure that was officially inaugurated in October last year, but it has a history already of underlying infrastructure systems that collect the data and made them available for probably 20 years. It all started out 25 years with the Asb Portal, which was basically a place where people reported bird observations, and from there it started growing and growing. And now we are here, and this is a very schematic overview of how the infrastructure works for the purpose of supporting researchers with data. So you have on the bottom, you see you have all sorts of activities, scientific activities, like people collect species, specimens in museums, they make observations as citizens, and these data are then captured by databases, and these databases, they are self-standing and pretty much independent, but they are connected. They're connected to a larger system here that harvests this data and then harmonizes these data in a larger data layer. And from there, anyone can access the data and then find also their way. If they want to have more information and need more source data, they can always trace it back to the original database. So the first thing as a researcher, what you can do is you can publish your data yourself. So here we have a lot of support functions to in terms of submission forms, even portals, software packages where you can actually add your data. But many people will be more interested in getting data out of the infrastructure. And there we have these three different interfaces that you can use. You have a machine-machine interface, or several actually, where you can use any sort of software to call the services that we have in infrastructure and download data or harvest data, is better to say. Then there is a human-machine interaction, which is a classic example is the normal portal where you can make queries yourself and download data. And then there's also a component we have in infrastructure with a human-human interaction that allows you to we call that the support center, to either ask us to help you finding the right data, or we organize training so everybody learns how to find and use data that are available out there. And of course, this system is connected to all the relevant systems outside of the country. So we have similar systems in every Nordic country, and they are very well connected, and also on a European level. Okay, so this is when we're looking at the conventional data, like basically species observation in time and space. We have about 100 million data records now. You see the different databases that give access to it, and you see it's very heavily unbalanced on the terrestrial side here. So so there is 100 million records, and now there is a lot of focus for the next years for us to boost these data records with this new kind of data sources that come from these machine-based observations. And one of them is the genetic data sources, of course. And here we have already a system online for genetic data, and it's called the Swedish Asb Portal. Especially it is designed for large data packages created by researchers and research networks on metabarcoding data. So what you can do here, you can have different functions. You see, you have a blast function, a filter function, and a submit function. So you can add data yourself, and we have all kind of support in place to help you with that. But
I don't know if you're
okay. That was GUI. Okay, GUI was found the mute button now. All right. So the other thing is the BLAST search, where you can actually submit any sequence. Actually, at this stage, it works for bacteria only, but during this year, we are planning to upgrade it so it works for eukaryotes as well. So you can submit sequences and blast them, and you will get a return on a map. So it's a geographic blast, and not only you will see the similar sequences on a map, but you will get, of course, all the source data and the metadata as well. Where did this sequence come from? Where was it observed? And all the associated sampling data with this. So you can also explore the database with filter function, where you can search for a certain gene, certain primers, certain taxa, and any other kind of details like expedition X, Y, Z, and these kinds of things. So that is these are the functions to allow to connect and integrate these new data with the conventional data on biodiversity. So that's one construction side, so to say, for our infrastructure to really make sure we're getting more data in. The other construction side is image data, and here we are now in the middle of building up a national system for image analysis just for the ocean, subsea image analysis, and here you see our infrastructure, which is really just taking the data products, which is when you come down to the point of you have a conventional species biodiversity data set, like species names, times, and geographic locations, then these data will then go into our system. But before that, in order to do that, you need all these different subsystems in place. Of course, you need the sensor infrastructure to collect the data to begin with, but then you need a data infrastructure to store the data, both for work as a workspace, but also for long-term storage. You need a place to analyze the data, and when you want to analyze these data with machine learning algorithms, you need to train these algorithms, so you need a platform for that. You can do that with citizen science, but you can also there's other places where you have reference image libraries that you can use. But once you have done that, you definitely need, in this case, also a high performance computing infrastructure that helps you running then these models across really large data sets. And all these components are actually in place today. They are connected, so they start to work. They need to grow, but they're already working in principle. And that will then, in the end, what will happen, what we expect to happen, that these type of analyses then create a large fraction of the marine data that we can then that help us then to understand what is out there and how is it changing. Okay, so these were basically the parts of the infrastructure that I wanted to show you. We have like already a lot of infrastructure in place, but we definitely need working very hard to upgrade our infrastructure so it can deal with these new types of data. And that is not a national process, it's an international process, so that every country is trying to do that, and these infrastructures, they talk to each other, they exchange data all the time. And just to round this up, I just want to go through a few examples of what these infrastructures can be used for now that we have access to these type of data, what kind of research you can do. I think my main point here is that you can go from hypothesis-driven research, you can add to that type of research with data-driven research. And that is really new, and that sort of research needs to have these infrastructures. Otherwise, it will not work. The difference is that in hypothesis-driven research, you would ask a question first, you define your question, then you go out and collect the data. But since there is so much data out there now, there has been added this new facet, which is called data-driven research, that allows you to look at data first and then pose your question afterwards. So these type of researches, they are sometimes a very good addition, and sometimes they cannot be replaced by hypothesis-driven research. For example, what you can do here is this one. That's already an older study. It was one of the first studies we did on the infrastructure, because when you have this legacy of data that goes back, when you're including museum data, you can go back 100 years and even more, and we explored that. So we had compared our own data against data from a reference collection from 100 years ago, and we saw that on the Swedish West Coast, where you see on the left side, these are the data points we could compare. It's like about twice as much species on the seafloor as today, or let's say as 20 years ago. So there was a massive loss of biodiversity on the seafloor. And what we found out in this study was that especially the rare species that disappear. So species that have not been very dominant to begin with, they were the first one to leave the ecosystem in the last century. But one of the things we could not answer here is like, how does a trend line look like? We only could compare 2 points of time. So this one is something you can do looking at historical data that the infrastructure gives you, but you can also look at systematic monitoring data, because these are also available by infrastructure. So we did a similar study, but with other sources, with other data sources, which come from SMHI, who did extensive monitoring programs in the last 30 years. And here we could get much more of a timeline. And you see the species diversity in these different parts of the Baltic and the West Coast, and you can see actually there is a slight trend that the diversity is actually seems to come back, especially in the last 15 years. I would say there is a slightly positive trend that biodiversity is actually recovering on the seafloor. But just to show you that these are the things you can really look at completely different time scales now. That was one. Another example is that we are able now to look at human pressures on marine ecosystems. So that was a master student project I had last year, and here we used basically satellite data to look at the species, but look at the human pressures. And what you see in the circles here, that is small boats that go in the summer period, they go into the bays and they drop their anchor very often. So what this student did, he tried to find out those hotspots where the anchoring pressure is extremely high, where the boats go over and over again, and he looked at how much do they overlap with the actual eelgrass places. So this is the biological data we could provide here, and you see the green parts of the map, the green areas, is where these eelgrass is documented, and the red spots are heat maps. The red squares show how strong the anchoring pressure is, how many boats you have there in the summer season. And on the left side, you see in an area on the west coast where there is a lot of pressure on an eelgrass bed, while on the right side there's very little pressure because the boats seem to anchor in a different place. There's no eelgrass. So these kind of investigations you can do with the data available through the infrastructure. And here's another example. We're now having also a backlog of 30 years in the Costa area of subsea images and movies, and we are training our machine learning models in the Costa Seafloor Observatory to recognize certain biological objects, to understand the process of change. So we use this to have currently we have quite a few projects on this one. Here's a few examples. That's one of the master students from this year who looked at the Bolosara, the deep-lead sea anemone, and he looked at the distribution of that species across time and over a larger time. Like, he's going through all the movies we have now. Well, he has finished that now, but in principle it's possible to go through the entire database and look for presences of certain species that we have trained a model for. So you can see variations in time and space in the abundance of certain species on the seafloor. That is another example. Here we have another example that we are currently engaged in. I see Francis is here online, so he is involved in this one. That's with the Swedish Geological Survey, where we're looking at various banks in the Baltic to train our models to recognize blue mussels abundances. So that helps to create very detailed habitat maps over large areas. So that is another application area. And finally, we have one here for, let's say, there is species that are very rare. So in this case, we have the cold water coral, Lophelia pertusa, or Desmophyllum as it is called now, where we do have observations and images, but they may not be enough because the species in Sweden, the species is very rare. So we have developed algorithms to create synthetic data. These are augmented images that are not real, but they still have the same properties as the original images in terms of the biological objects they contain. And these ones then can be added to the training data and then improve the model. So we have right now obtained with that model, with that method, a very good model to recognize cold water corals in Swedish waters. So finally, about the genetic data, there is another example that we are currently using. The genetic data that become available through this infrastructure now. They can be used to do establish a monitoring system, and we are especially interested in monitoring rare species and invasive species, or let's say alien species, because not every alien species will become an invasive species. But you see here where we have these genetic data, they are coming together through the infrastructure and become accessible. And just from 1 year, we can find 35 alien species that some of them, they were known to be alien, but many of them, they were actually new. They have not been discovered here before, while some of them, they have been discovered many, many years ago, as far back as in the 1920s. So they're very rare species. And this data now, when you're putting this genetic sensing data together with the conventional data we already have from museums and from systematic observations, then we think we have enough data to do modeling and forecasting, so we can start using these data to build distribution models that make prediction on where these species are aggregating, where these species are kind of assembling in as hotspots and where they are likely to pop up next. So this is kind of a biological weather forecast we are designing here now, which is only possible if you have really large volumes of data available to find out those species that you are interested in, to get enough background data for the species that you're interested in. In this case, it's alien species. And I think that is everything I wanted to say. So I thank you very much for your attention. And if you have any questions, let's go.
Thank you, Matthias, for the very interesting and very informative sharing. And yeah, we welcome questions from the audience. You can directly turn on your mic and ask, or you can type your question in chat.
What are the biggest challenges now?
That was David, was it?
Yeah, that was David.
Okay, yes, there's lots of challenges, but that's good. There's of course a lot of technical challenges, of course, and I would say that they have to do with the data management per se. You have to get the data never really in one place. So the thing we want to learn from, for example, the community, we want to learn from is the satellite community. They have sorted it all out to a large extent, and that is because there is often the source of the data is already is either one satellite, often is one satellite that collects the data, and then they're in the right format for a large area or for a long time, and they don't need to be these patches don't need to come together, need to be integrated. So for biology, the problem with satellite data is it's not telling you, I mean, it's not explicit about so many species. It's just often biological patterns, but not necessarily species. That's one thing. You can't observe awful lot of species with satellites, but when you want to observe species, often you need a distributed infrastructure. You need a sampling in different places by different people under different conditions, and that leads automatically to the source data will be in different places, and with that comes all the problems of trying to patch them together afterwards. So that is the main problem. The bigger the data get, but they come from different sources, to make sure they come together, they're harmonized, and they are also collected according to standards. And that it is a big advantage in that challenge that there are machines that do this, because it's easier to program a machine compared to programming a human. It's possible to program humans, but it's much easier with a machine. Yeah.
Okay.
We got a question from Yan. How about data-driven management planning? Is the data ready to use in large-scale management? Which gaps do you see to address in future work, both with regard to the infrastructure and the data?
I think the data are used a lot for management right now, but since there is so little marine data in the infrastructure at this stage, compared to, there is a lot of data. When you're looking at SMHI, it's really the major provider of marine data, but compared to the terrestrial data, is a minor fraction, I would say. So as this data is starting to grow, the applications in management will start to flourish and to diversify, and will be used for all kinds of different management purposes. But the data publication thing is something we have to focus on. because we had so far in the last 10 years, we looked only at publishing big data sets, let's say from the Museum of Lund or from the Museum of Gothenburg. So these data were already very well prepared, very well organized, and then they could be connected to the system. Now that we've done that, we have connected most of the large biodiversity data providers in the country. Now the challenge is to go for the medium-sized data providers. That's like large scientific networks, anyone who's sitting on a lot of data and they are not available yet. And if these data are organized and they are according to a standard, then it's pretty straightforward to do that. But most data aren't. They are usually messy, and that's natural because biology itself is very messy. So we're now working to capture these medium-sized data sets.
Thank you. Before I ask Frida's question, I just want to follow up on this. Matthias, you mentioned that biology is messy and also sometimes the data can be messy. So does that mean like new standards, when you import some new data, then you also have to revise the standards? Or
Yes, for some of these new data, the standards are only just now coming together. So for both for the genetic and also for the image analysis, which I think are the ones, the data source with the highest potential to really create huge data sets, very detailed data sets, very effective, very cost effective. But these standards are only starting to develop right now. In genetics, it's a little bit further, but the genetic itself, the genetic information is very well standardized, but the metadata around it there's still some work to do, and that's a very intense process going on everywhere now to make sure that these standards are there, they are implemented, they're used. So one typical thing is like researchers and research agencies, when they supply money now, when they fund somebody, they make sure that there is a data management plan in place and it's also followed up. It's not just written, but it's also used. And these kind of, it's a different culture a little bit that is now coming more and more and is becoming actually, yeah, I have a good feeling about it, that marine biologists, they actually adopt this and they see the benefits of it, they see meaning of it.
Thank you. And Frida's question, what kind of data could be helpful to collect through citizen science? What are the current challenges in that kind of data collection?
There's, citizen science. So we're using citizen science, but I wouldn't call myself as expert in citizen science per se. But there's a lot of really high potential. We see that in citizen science using citizens. There's all these different facets you can achieve with citizen science, like people make more literate about the environment and the environmental problems, and that you can actually, they can collect data for you. And there's a third one. I think there was always this thing that citizens should somehow by participating, like this kind of participative science, they should justify or legitimize what we are doing for them. But now I think that it's changing, the idea that the citizens actually, they need to contribute. Just that everyone understands that these researchers should do this environmental science is not enough anymore. Everyone needs to participate, because those researchers we have in each country, they're not enough. They're not going to be enough to collect the data. So we need citizens and we need machines to deploy both citizens and machines, and there are standards coming together very fast. The social scientists are really leading the frontier, and we are learning a lot from them. And then I think it can be very powerful to deploy citizen science, because you're achieving so many things at the same time. So what we see is that citizens, they become much more aware, and that affects them in their everyday life, and they are much more connected also to the scientific process, the way we see at the environmental issues and what we're trying to do about it. So it's not anymore the citizens that talk to the politicians and deliver their fancy reports, and then the politicians go to the normal people and say, like, yeah, the citizens told us this and that, now we have to implement this and these laws. It's all coming together. So the citizens are already on board. They understand that before even the politicians will start talking about it, because they have been creating the very data that helped making these reports, something like that. So I think that's a power that is a much wider concept of citizen science than just citizen science boosting our data results. So basically helping us creating more data.
Thank you, Matthias. And another question from Jonas. How can we encourage people to upload on old or historical data sets? Is that even useful, as the metadata is often missing or flawed?
That's a very good question. The thing is that there has been in the beginning in this digitization initiatives, and I think 10 years ago, there was so much data out there that was valuable, and we thought like this community came together to capture it. And then you come across some data sets where you think, like, is this really worth capturing, or is it they're not going to say anything, or it's so difficult to digitize it and capture it? So you may as well wait for the for a new method that comes along to create that data from scratch. So I think there has to be in every case a decision on what is the potential value of the data, and are we going to be able to collect it, capture it, and then also maintain it. So that is a very common thing for museums or any other infrastructure. Like when museums, they have to, when they get objects delivered from the public, they have to make that decision every time. They have to say, like, this is a really valuable specimen, and it's worth keeping it here in the house, and with all the resources it's going to cost to maintain it for the next 100+ years. And some of them, they will be obvious, it's like, no, it's not worth it. And it should be the same with data. So we could look at the data, say, like, what is how valuable are these data, and then is it worth to maintain it and to keep it, because it's not for free. It costs money to maintain data. It's not just putting it in a hard drive and putting it in the basement somewhere. I think that is my answer.
Thanks. And another question from Bruce. And how do you propose to handle the uncertainty on the collected data?
Uncertainty, yeah, that's like when you say, I guess you could say you mean the quality of the data. So the quality is not necessarily an issue, because there's all kinds of ways to check the quality, and for the question at hand, you only need to have a certain quality. So if you change that question, if you have another question on the same data, you may have completely different quality criteria. So the important thing is that you can apply these criteria to sort out the data that you want from any of the data you got. If you citizen science data, genetic data, if you go on GeneBank, it's full of trash. but you don't need to use it. Same with this citizen science data. There's a large portion that is not valuable, but it's not making any harm. You can always filter out the data you want at any quality level you decide you want for that question at hand.
Thank you. Yeah. So I have a question that regarding the usage of or access of the data. So do the users have to be affiliated with some research institutions in order to use the data in the Swedish biodiversity data infrastructure? Or is that, or are you considering like open access, or even maybe collaborating with some platform to create open challenges for people just to freely explore the data and see what they find out?
Yes, it's a very good question. The data are open to begin with, all of them, but there is some limitations with sensitive data, and that applies to, for example, red listed species or sensitive species and places that we cannot disclose for various reasons. And these data in the conventional data set, there's 100 million records. I think it's 1 million records that are not open to the public. But here again, it's not only either it's open or it's closed. There's many levels of, like, when you're a scientist or when you are a person of a certain institution, then you will have access. So it's a gradual change in access to certain data, but it's a very minimal part. About the subsea data, for example, there is like depth locations and also geographic information is not accessible. It's not allowed. It's not legal to publish these. So these data of subsea images, they will never have any associations with geographic coordinates, and probably also not depth.
Thank you. So there are, in other terms, there are various concerns or constraints regarding certain data.
Yes, but they don't restrict the data as such, because data is not always one value. It's often a combination of values, like you have a measurement and you have a species name, and some of these values, they will be masked. They will not be available under some circumstances. So it's not that you have to restrict the data itself, you just restrict certain values under certain conditions.
Okay, so it's like maybe certain columns or certain.
Yes, exactly.
Yeah. That's good to know. Yeah. And so does the audience have any question?
I have one question, if you don't mind.
Please.
Okay, good. Nice, thank you. I have appreciated the talk also because I have helped a little bit developing a pipeline in SBDI to submit molecular data. It is called AmpliSec.
Ah, okay, great.
Yeah. So I was working for the microbiological molecular part.
Yes.
Which became the molecular module of SBDI. Yeah. But it was a small contribution. I need to be honest about it. But it was nice to see how SBDI is coming along with the other institution to join forces. That is nice. And I wanted to ask if right now the molecular module is also ready to take on genetic data from eukaryotes, not only bacteria. Do you happen to know something about it?
Well, I'm not completely updated. My update is like 1 month old, but we had it when we launched it. We had it online. We had 18S, I think, and ITS.
Yeah, because it started. We have also collaborated with. I was working in LNU, so Linnaeus University, but together with both Stockholm, the group of Anders Andersson.
Yes, they're leading this. Yeah, they're leading this.
Oh, yeah, of course. But there was also the group from Uppsala, which was working on the fungi, and this is where the 18S sequencing sequences came from. So yeah, I should have
Yeah,
been precise.
but my suspicion is that it's somewhere there in the background. They probably took it off for now. What I remember is that it's not so trivial, and that's where these infrastructure, when they come together, the genetic one, it has a different taxonomic backbone because it's the one from GeneBank, and the other taxonomic backbone from all the conventional biodiversity data will be Dyntaxa, and for the marine world it's called the World Register of Marine Species. So that is something where I see the large difference, where this is much more difficult for the biology compared to physico-chemical data, because there it's temperature is always temperature, and you measure it always in degrees Celsius, maybe in Fahrenheit, but it's not more complex than that. But here in biology you have a species which changes name, and every species has like 7 or 8 on average synonyms. So what you call species A in one place, you call it species B in another place. And then there's all these changes that come when you do taxonomic revisions, that names can change from one to another. And it makes things awfully complicated, especially if you have different taxonomic reference systems. So you have to make sure that the biological entity is the same, even though the name is not the same, and the other way around, that the name is the same, but the biological entity is not the same for some reason. And that is when you're measuring biological entities with genetic data is a different taxonomic reference. And when you're measuring it with, basically you take an Excel sheet, you're noting down the name of a species that you see for the microscope. That's what you did in the last 100 years, and now you are relying on genetic data. So these reference systems, they have to talk to each other, and it's not done overnight, but it's coming together. So to answer the short answer to your question is, it's definitely coming this year, eukaryotes.
Okay. Yeah, I mean,
I
like
guess
animals.
before the summer, yeah.
Yeah, I was more referring about animals because, as you correctly pointed out, fungi, they have already been taken care of. But yeah. I know it can get complicated. It's a matter mainly of reference databases when it comes to the annotation, as you correctly said. And I remember the pipeline. I think by now it should allow to at least choose or import the reference database for the annotation. Not sure, but and then the other thing, as you said, is the having a common backbone. The guys from Anders lab, they told me I think they're still struggling with having a common backbone with the GBIF database.
Yeah, exactly.
So but that was very cool. It was very nice hearing all of this from you. Appreciate it.
And thanks for contributing. And like we definitely plan to have, for the genetic parts and also for the image analysis part, these new things. We definitely try like to organize workshops. We have material already out on the net. So if you go on the website, there's YouTube videos, tutorials, documentation. So if you want to publish data, if you want to use data, it's all there. But then of course we have to update it, and we have to get also closer contact with the researchers to have like data publication workshops or these kind of things, where everyone comes with their data set and we go through it and show them how to publish the data. That's my vision for the next 2 years, that the infrastructure makes really a lot of personal contacts through workshops with users.
Okay, then I'm looking forward to seeing all of that. Nice, thank you.
Thank you.
Great. Then we have some already potential data contributors. Yeah.
Great.
Yeah. Do we have any other questions or comment or sharing? Or maybe have you thought about like Matthias, you shared about the doing research from the perspective of hypothesis testing and now to data-driven type of research. So what do you see are the kind of differences or in such kind of mindset when it becomes more data-driven?
Difference, yeah. The difference is that most research, because these infrastructures are only coming together, most people are not aware of the opportunities for data-driven research. So when people think data-driven research, they often think like, oh, I have a colleague with a big data set, or I have a big data set myself. And I think you always, in the normal research cycle, you would always collect your own data, but you should much more enrich your own data with the data that are out there. And that process is not so well established, I think. So it's not one or the other. It's more like seeing this as a new opportunity to improve one's own research profile, to say, well, yes, I am interested in this, I'm investigating this ecosystem, but I'm very much aware of the data and the background of knowledge that's out there in form of data methods. So I go there first, then I go out to collect my own data, and then I have a really massive data set. For us, for example, the classical examples, we work with invasive species in the area, so we make genetic observation of alien species, but we only know it's an alien species if we know where it comes from, and we need all that global background data. We need to connect to these systems that have observations of species outside Sweden, anywhere else. And without that, we will know very little about like what's the dynamics behind this migration of the species to our waters and these kind of things. So it's really important to be aware of the data sources you have out there, and also the tools that you can use to explore these data sources. And I think most marine biologists that I know from my colleagues, I don't see any. I mean, they are in the middle of it. So it's just really just informing everybody about the upgrade. Now we have a genetic database, and the image analysis is coming next year. You have this tool and you have this database, and providing documentation, very good documentation, contents, and workshops, training events, these kind of things. Then it's fine.
Thank you very much. So it will be a learning opportunity also.
Oh yeah.
Great. Okay. So do we have any other question or comment? So if not, then I think that's the end of the webinar today. And thank you all for coming. And thank you so much, Matthias, for sharing with us. Thank you.
Thank you. I'll see you on the next webinar.
Yes.
Thank you, Matthias, Isin, and everyone.
Thank you.
See you next time.
Thank you, everyone. See you next time.
Bye-bye.
Bye. Take care.